So, welcome everybody to this new season of EBD seminars. As you can see, the organizer committee has changed. Maria and Javi did a wonderful job, and we hope to do our best. John is one of the organizers of the EBD seminars. Katarina Ruthschat should join us also the next month. And myself, Isabel Salado, we are going to present the seminars in the seminar starts. We encourage PhD students to present the PhD plan during the first year, as you remember from previous years. Also, one of the we are going to include another kind of seminar that is going to be more related with outreach and will be in Spanish. And as it used to be, the last day of the month, it will be focused for discussion group. So the next week we will have a discussion group. So I will introduce you the next speaker. That he is Ruben Evens. He is an assistant professor in the University Catholic of Leuven in Belgium, and he's a behavioral ecologist, and I think he will explain very interesting things about nightjars. So thank you for coming, and the floor is yours, Ruben.
First of all, thank you very much for having me. I've been here 10 years ago when I was a bit younger, and Carlos was also a bit younger. Carlos and I, we met in 2010 as two very young nightjar enthusiastic people, and our dream was to study nightjars. So here we are today, 14 years later, and I'm very happy to be here. So my talk today, I will try to summarize 15 years of research in 40 minutes. I will do this by very briefly introducing myself, showing how the study started. We'll talk a very tiny bit about nightjars. What are they, especially the European nightjar? And then I will highlight some of the methods that we use during our research. And then at the end, I will give an overview of some things that we studied in the past. And we will look also in the future, or to future ideas that we are currently working on. So we'll talk a bit about space use and habitat selection. I'll not talk about migration today because that is the work of my PhD student, Michiel. And then, as you saw, the title nights. So in the last couple of years, our research, the more conservation-based research or focused research, has evolved into a more study on the behavior of individuals, and especially in response to the light environment. So that is what we will talk about a lot at the end of the presentation. So this is how it started, 2009. I was a slightly naive master's student at the university, and Eddie Ulenaars, who is sitting there, he's a forester in this part of Belgium. And we had a bright idea of studying nightjars. Many years later, I did my PhD at the University of Hasselt, where we tried to focus on evidence-based species conservation, trying to restore habitats of the species in Belgium. And then I migrated myself to the Swiss Ornithological Institute, where we also performed evidence-based species conservation studies to track how nightjars would live within these in the Swiss Alps. And then we go to 2019, just before COVID. I had the opportunity to work at the Max Planck Institute with Bart Kempenaars, and there we evolved the idea to work on light-mediated behavior of the European nightjar. Unfortunately, COVID happened, so I was working at home and not physically at the institute itself. But there, all the concepts and ideas evolved to then get my FWO postdoc in Belgium, where we worked on the impact of skyglow on the behavior of this European nightjar. But more on that later in the presentation. I always like side projects. So we have some side projects, or we had some side projects in Africa as well. So where we went to Ethiopia to study nightjar diversity in Neshisar National Park. And in the last years, we are trying to set up research in the Democratic Republic of Congo. Not only because our Belgian nightjars winter in this area, but also because we are trying to set up projects with local scientists, such as this guy, this Papi Dunia, and together with him, we are currently setting up a project to study local nightjar diversity in Congo, and he's looking for this nightjar species. It's the Itombwe nightjar, and it's a species that is only known from one individual in a museum collection. So he's trying to find a living or evidence that this species is still living in the Itombwe forest. All right, let's go to the most important species of today, the European nightjar. For those who do not know, the European nightjar has a Eurasian distribution, and it has been suggested there are 6 subspecies. Here in Spain, to a certain extent, we have the Meridionalis. And if you go on Wikipedia nowadays and you look for where are the overwintering areas of this species, then you will still find it's either in West or Eastern Africa. And in a few slides, we will see if that is true or not. So the annual cycle of this European nightjar. So currently we are in September, so our birds are now migrating towards the wintering areas where they arrive at the end of October, early November. Then they overwinter there and they start departure somewhere between February and March. So they can arrive at the beginning of May in the West European breeding sites where we study them. And then they have a breeding season until the end of September. We did some studies on the migration of the species. Again, I'm not going to elaborate a lot on this. With colleagues in the UK and in Belgium. So our birds, they cross the Mediterranean in spring. They move to the wintering areas below the tropical rainforest. And then in spring, they go to Western Africa before coming back. And many of the birds then use this area to go back to the Eurasian wintering areas. In black, you see geolocator tracks, which are less accurate. And you see we studied also some birds in Mongolia to see how they migrated towards the African wintering sites. The European nightjar lives in a completely different area or habitat, more or less, than the Spanish redneck nightjars do. So for many years it was believed that the European nightjar is a typical heathland species, and they would need nothing less than a nice patch of heathland, either within a forest complex or just within a very vast heathland area. And this is one of the reasons why we started the research in 2009, trying to understand how we could conserve or restore habitats like this in order to increase the nightjar population in Belgium. So again, this is, for example, 5 hectares more or less, and it was believed that one nightjar territory or one nightjar couple would just need 5 hectares of this, and they would need nothing less. They would eat here, forage here, rest, sleep, and reproduce. Talking about reproduction, nightjars are a very interesting species when it comes to studying their nesting behavior. They don't make any nests, so they find a suitable spot somewhere in the heathland in our case, and they lay 2 eggs. And that they then, the female incubates, and the male on some occasions helps her with incubation and rearing the chicks. Nightjars, they can have 2 clutches, or with us in Belgium, they can have 2 clutches in 1 year, so a maximum 4 chicks that reproduce or that are raised per season. Something that I always find most important when I speak to people about nightjars is that they understand how these nightjars forage or how they live at night. So a nightjar is a crepuscular species, which means that they are mainly active during dusk and dawn, so when there is still some daylight available, and how this translates into the nocturnal activity later. But what is important is that one of the main foraging tactics of the European nightjar is a sit and wait foraging tactic. So they sit on a pole or a branch or a tree, and not so much on the ground as the redneck nightjars do. But they sit there and they wait until they see a dark flying insect against a bright sky. And what they then do is they perform a parabolic flight to catch this insect, and they go back to the perch. So they actually need some sort of light, some light background to highlight this dark object in order that they can forage and be active. So nightjars are insectivores, which means the European nightjar at least mainly eats flying moths and beetles to a lesser extent. All right, so how do we perform this work? Our main study sites are in the northeastern part of Belgium. So this is the province of Limburg, this is the province of Antwerp. These are sandy soils, and on these sandy soils you have the heathlands. And we perform our studies in the big strongholds of the species in Flanders. This is the northern part, the Dutch-speaking part of Belgium. In the southern part of Belgium, so in the French-speaking part, you have a few small populations. Those populations are decreasing rapidly, and at the end of the study I will tell you why I'm interested in these populations, why I want to start studying these as well. In contrast to the work of Carlos, who can drive around in his van and catch all the nightjars he sees, we have to put up big nets in the territories of our nightjars, and we have to try to catch them with these big mist nets and tape lures. Yeah, that was a spider. Yeah. So we extract these birds from the net, and then we take biometric measurements of each individual. So we age them, we sex them, and from each individual we also take a blood sample, because we do quite some parental analysis, extra pair paternity, for example. So we have at this moment a data set of approximately 1,500 individuals that we sampled, and that we know relationships between all these individuals. And then at some point during the processing, we can decide to put tracking devices on them, because our study mainly focuses on understanding individual behavior. The most basic technique is putting a radio tag on the bird with superglue. So it's a very simple device that emits a signal that you can receive with an antenna. So you can more or less triangulate the position of this bird. And then, as I will show you later, we can also use a combination of a radio that emits a signal, a GPS and an accelerometer, but I will show you more about this later. And we also deploy quite some migration loggers to understand how these birds behave during the season, but also during the wintering, and how they go to the wintering sites. So already told you about the radio telemetry. That's how all our work started in 2009. It's very labor intensive, because are a few sparse observations of where these individuals are within the breeding sites and in the foraging sites, for example, here in green. And what happens in between is very vague. The amount of data that you can collect per individual is quite low. And that's why why technology luckily evolved, and why we are starting now to use different types of loggers to understand individual behavior of the species. So nowadays we use a combination, as I told, of a radio tag, a GPS logger, and an accelerometer. This is still the old version. Nowadays the accelerometer is integrated in the GPS logger, so you just have this device and the radio tag. We deploy this device using a water-dissolvable string. So in Belgium, in contrast to Spain, it rains a lot. So the loggers drop from the bird after a few days when it rains, and we just have to go into the field to collect the loggers. So we don't have to recover or recapture the birds. All our devices are archival, which means that all the data is stored on the logger itself. So we need a logger in order to get the data. But because the loggers drop from the birds, we can just collect them in the field, and we don't need the individual in our hands. That's the only function for the radio tag so far. So the GPS logger allows us to collect information about the location of a nightjar every 3 minutes during a period of approximately 10 days. And evidently this gives us more information about how long an individual stays where, how it connects different areas. But at this point we were still a bit disappointed, because we could not really pinpoint or understand what individuals were doing. We could guess it. For example, an individual that is sitting in a breeding site, it could be singing, it could be resting, it could be incubating, it could be eating, foraging. We actually didn't know. The same for birds that are in the foraging site. Of course, we can assume that these birds are feeding, but are they maybe also singing, etc.? We didn't know. And for that reason, we started to use accelerometers a few years ago. This is not the accelerometer we use, luckily, but this is the concept. So an accelerometer measures body acceleration in 3 dimensions at a frequency of 25 hertz. So 25 times per second it measures body acceleration of the animal. Here you see a representation in one axis, and when this person pushes the cell phone, you see that there is a peak in acceleration in a certain axis, and it is registered by this device. And that's the same way that our devices also work on these nightjars. So instead of having purely information about the whereabouts of an individual, we can now understand how long it is singing somewhere, how much it is eating, etc. I included one small example. For example, this, so we can identify 4 types of behaviors. I will talk about the singing later, and then we have the resting and flight activity. But if you see at the leaping, so that's an individual sitting, flying towards an insect, taking it and going back, or at least attempting to catch an insect. We don't know if they really eat it at that point. So here we see the bird is resting in blue, and then suddenly it performs a very strong wing beat, and then it glides back to the post. And this way we can actually count how many times an insect attempts to, a nightjar attempts to eat an insect during a certain period of time. Yeah, I have a small video of it. So this is a nightjar sitting here, flying towards an insect and taking it back. So this is the type of behavior that we can, or this is the way that we try to understand how our birds behave at night. This is more or less, in a very small nutshell, what we try to understand about the individuals. But what has become more important in the recent years is also trying to understand the environment, the light environment that our birds live in. And for that reason, we use sky quality meters. So these sky quality meters are small devices that we place in our study sites, and they measure a small fraction of the atmosphere every 3 minutes. And this gives us an understanding or an information about how bright this section of the atmosphere is. So to give you an example, when this device measures during a full moon, then you would expect a value of 17 magnitude per arc second, which means the sky is very bright. There is a lot of light in the atmosphere. When there is a new moon under natural circumstances, we would expect that the sky is very dark. So then it's a value of approximately 20 magnitude per arc seconds. And under natural circumstances, when it's an overcast night, you would also expect that atmospheric light, so for example, starlight is blocked by the clouds, and then you would reach values that are even darker, around 21 magnitude per arc second. But then, of course, in Belgium, we don't have natural circumstances anymore. So what happens in Belgium is that during these new moon nights that are overcast, we actually started measuring sky brightness that is equal or even brighter than full moon nights. So for us, this was a very interesting case to understand how these very light-sensitive species, or this very light-sensitive species, would adapt its behavior and how these individuals would react to such rapidly changing light environments. The only disadvantage of the sky quality meters is that they just give information on a small section of the nocturnal sky. And also it doesn't tell us whether what is measured is moonlight or, for example, skyglow. And for that reason, we started to use all-sky photography. So all-sky photography is a very standardized way of measuring light in the atmosphere. We take pictures on a very standardized way under different environmental conditions, and all the settings in the camera, a calibrated camera, are also standardized. So it's always the same conditions that the picture is taken in. And then we get stuff like this. So this is one study site in Belgium, just to give you an example. Let's start here. This is a clear night with a moon. So you see the atmosphere is very bright. And this is the same location, but without moonlight and a tiny bit of clouds. And then you suddenly see here a very bright spot of the harbor of Antwerp, which is approximately 15 to 20 kilometers further, but it brightens the entire horizon. And then you have a small amount of municipalities here. And then you have this site, but under new moon, so no moonlight, but with cloud cover. And then you see how it brightens the study site. Again, this is 20 kilometers from the closest street light. So then you might wonder, why do you have this ugly formation of your pictures? Well, because this is a sky in Granadilla in Spain. And then if you then understand how dark a new moon night should be, then my question is, how can this affect the behavior of a very light sensitive species? So for this was for us a very strong eye-opener to start to understand how a nocturnal species would be affected by skyglow. So the light that is from a very distant but still affects the nocturnal light conditions in a certain site. Of course, we don't use these unprocessed images, but we can process them. So these are the same conditions. So new moon with cloud cover and a full moon. And this allows us to understand where the light comes from and how bright every section of the nocturnal sky is under different light conditions. So, for example, yeah, this is a new moon, but you see that there is quite some light pollution on the horizon. And then this night is even brighter than, for example, a full moon night. So especially the zenith is very bright. These are the light conditions. And for example, this night is approximately 500 times brighter than under natural circumstances. At this point, to be honest, we don't know how it impacts the birds yet, or we are trying to understand it. But this is research that we started a few years ago. This is the picture. These are the light conditions. And it's something that we completely do not understand at this moment, is how sky color could be affected or could affect individual behavior. So this is showing the color temperature of a night. Let's start with the easy part. This is a full moon, and then you see the sky is very blue. But if you start to take away the moon, then suddenly your sky changes, but you see the effect of potassium lights, sodium lights, so the typical lights that are used in industrial areas, for example. You see that they make the sky more orange. But again, that is something we, at this point, don't understand. So if anyone has ideas about this, I'm very happy to discuss it. All right, let's close the methods section, and let's start looking at some research that we've done over the past years. Again, let's fly back in time. 2009, we tried to understand how these nightjars live in these very small habitat patches, these very fragmented heathlands in Belgium. So we deployed 150 loggers, radio tags to run around as crazy to find how these birds live. So in red, you see the areas where these birds were tagged, more or less the breeding sites of these birds, and it was expected that they would nicely fit or stay within these red areas. So and then what happened is that, so these are all the data of all the individuals that we collected over these 4 to 5 years, and one color represents all the individuals of 1 year. If you see differences in color shifts, that's because we just tagged different birds in different areas. But the take-home message here is that these nightjars actually didn't really like the breeding sites at night. So what they did is they woke up and they went outside of these breeding sites to go elsewhere. So you see some birds going here, here outside in these areas. You see some birds going here, but yeah, for us the most important area was this. So this is a valley of a small creek with a lot of extensively grazed meadows, etc., where these birds apparently went at night. If we did some analysis on the home ranges of these birds, the areas that they use, so this is just an example of 4 individuals, but we did this for all the rest of the birds as well. But it went up to 125 hectares or even 2,000 hectares for some individuals, and with foraging distances up to 2.5 kilometers using the radio tag information. If we then looked where that was especially to these extensively grazed meadows with a lot of herbs, and the reason why these birds went there was actually also investigated. So for 3 years we deployed light traps in different habitats, and we found that in these breeding sites there was at least half of the amount of food available than in the foraging sites where these birds went at night. And it was not only the abundance of food why they went there, but we did some metabarcoding on fecal samples, and we found out that these birds, they use a particular group of insects, not specific species, but moths with a wing length of approximately 22 millimeters. And those are also the group of insect that is mostly found in the foraging sites and not in the breeding sites. You see this very high abundance of Arctidae. Those are I think they call the bear butterflies, and they are very, they have a lot of scales, and the birds don't really like to eat them apparently. So in 2016 we started to deploy loggers on these birds, and meanwhile I think we have more than 300 individuals that we tracked. A very quick example is how we studied these birds in Switzerland. So this is a study site in red, where a previous study using telemetry showed that the birds would not leave this breeding site, but we tagged every male in the population, and all the birds actually moved outside of the breeding sites at night to go into alpine meadows. And very interesting was, for those that among you that know Switzerland, the valley where we studied this is the Wallis region, so the valley of the Rhone. They make a lot of wine, and our birds, they selected biologically managed vineyards. So winemaking is they really like to use pesticides. Let's put it that way. But the birds really knew where the biological vineyards were, and they went there to find their food. All right, let's go to chapter of the day. So during our work in Switzerland, I was intrigued by how the river or the valley of the Rhone looked like. So you have these municipalities, these towns, and if you look into the valley, then you start to see this very nice glow. So this is a nice example of the difference between direct light pollution. So a street light might have a local impact on everything that lives around the lamp very locally, but a fraction of this light is also projected into the atmosphere and creates this glow. And this is the sky glow that I'm interested in. But to start at the beginning, we were curious to find out how moonlight would affect individual activity. And that's why we started to use multi-sensor loggers. So these multi-sensor loggers, among things, they measure sunlight and flight activity. And using sunlight, we can geolocate the position of the bird on Earth, so we know whether it's in Belgium breeding, whether it's flying through Spain to go to the wintering areas in Africa, etc. And then it also measures flight activity. So every 5 minutes, we know how much the individual has flown in this 5-minute time frame. And then you can make these nice ectograms. For example, this is an ectogram of one individual during one lunar cycle when it's wintering in Africa. And what you see on a horizontal line is 1 day with local midnight in the center. So this is the afternoon, this is the evening, this is the night, the morning, and then the time before noon. and the height of a bar indicates how much an individual has flown in a 5-minute interval. So what you hopefully see is that these birds, they really like to be active in the evening, and that they are also active during certain parts of the morning, sometimes, not always. And then hopefully I see that there is a nice diagonal activity pattern, especially around full moon. And what this corresponds to is actually the lunar cycle, the local lunar cycle. So during a new moon, there is no moon at night, so these birds are barely active. During a full moon, then the moon is above the horizon the whole night, and you see this nice activity pattern at night. And then during the first and the last quarter, you see that the moon is only above the horizon during one part of the night, and this also corresponds to the moment that these birds become are active or become active again. Very logical, you might think, but then I started looking at some data from Mongolia. And so these are two birds that are in the middle of their breeding season in Mongolia. And then you might notice that there is suddenly a drop of activity around full moon. And I was very curious to find out what it was. It's very anecdotal. But then I started looking up some weather data. And this was actually during a period of heavy overcast, so there was no rain expected, but it was very overcast. So apparently these clouds might have blocked moonlight, and these birds became less active. But then I was wondering, I have never seen a dark night in Belgium. So then I started wondering, so this is one of our study sites. There is nothing enhanced, this is just a video. How can this skyglow affect individual activity? So we started to look at these flight activity data again, but instead of looking through the whole year data, we started to compare data from Belgium and Mongolia. So we have individuals that are tagged in Belgium. We started looking at the breeding data. We have some birds in Mongolia, and all our birds, they winter in the same more or less dark area in Africa. So we were able to compare their activity patterns during the summer and the winter, accounting for behavioral differences due to breeding sites, for example. To cut the story short, we noticed that our birds in Belgium, they actually can choose whenever they become active, so they don't follow the lunar cycle that strongly anymore. So as long as the more heavy the overcast night was, the more active these birds became, suggesting that they are not learned very quickly, or they're plastic, I don't know, to exploit these overcast nights and become active, fly more than you would expect under natural circumstances. So at this point, we believe that the skyglow in Belgium has the potential to relieve our individuals from visual constraints that they would have during a normal or under natural circumstances. And the next goal is to really start looking at individual behaviors. So how much do they forage, fly, sing, using these three-dimensional accelerometers. And for this, we are currently trying to collect more and better data on individual space use and behavior, but also on the environments, as I showed during the method section of this presentation. So one of the things that we are working on and discussed with Carlos that I quickly need to finish finally is data that we looked at using the GPS accelerometers. So we know every 3 minutes of the night where an individual is, but we can also count the number of times they attempt to catch an insect. And from this we learned that individuals that, for example, forage in brighter environments, they also attempt more fly-catching events during 5-minute intervals. So if we look at these foraging data, then we see, okay, if you're foraging or breeding in a bright area, then you're more likely to eat, or at least attempt to eat more insects at night. So this is the first insight that we are now getting into these really specific behaviors and how they affect individual behavior. So while I'm transferring to this new position, I'm lucky to have also future perspectives, meaning that we are starting up studies with new PhD students that, for example, will work on Nightjar communication and how it might be affected by skyglow. We have a master's student that looked at how individuals might perceive skyglow while foraging, and the same master's student will become a PhD student in a few weeks and will start looking or starting up a study on juvenile dispersal. So what I didn't tell you at the start of the presentation is that in the European Nightjar, only the male has these white patches. So they have 3 white patches in the wings, or these subterminal patches, and you might see it. They also have the terminal white patches on the 2 outermost tail feathers. So one of the things that Richard is going to investigate is how skyglow might affect the size, or how these birds can use, or how the communication, the social communication with these white patches is influenced by the presence of light. Among things, there is then the natural light and skyglow, of course. Another way that Nightjars communicate is by singing. So we can use our accelerometers here to identify when individuals are singing. So this is a study that we did some years ago as a trial to see if we could identify song activity from the accelerometers. This is the accelerometer track. In red you see song activity, and in this sonogram you see when song activity was recorded by a device that was placed in the territory of a bird. And as you see, these nicely match when the bird is singing close to the to the song meter. But then we also had these intervals where we recorded song activity, but then on the GPS data we could see that the bird was not in proximity of this this logger. So now we are going to use all the data that we have on song activity of males to see how the lunar cycle, but also the breeding season, mating status, etc., and also skyglow might affect how much and when and where these individuals are singing. I told you in the beginning of the presentation that these nightjars, they like to perch and try to use a bright background to identify prey. Well, a study that this master's student did was actually look at GPS data combined with magnetic data to try to understand how nightjars position themselves towards bright backgrounds. To give you an example, so this is a foraging spot of one individual during a few hours. This is a high tree, and the bird is using some perches to sit and to see for look for prey. So what we know at this point is, okay, if this bird is sitting here, how is it looking, or what is it doing actually? And then we have information on the activity of the bird, so that's the overall dynamic body acceleration, so we know whether a bird is sitting or whether it's performing very short, quick wing beats. For example, this is a flycatch. So this is a flycatch with a longer intermediate flight, another flycatch, etc. So the bird is sitting still. When the bird is doing nothing, it is looking in approximately 200 degrees. So the bird is looking in this direction, so just next to the tree. If we go a bit further on here, the bird is sitting and it's looking in 100 degrees, so it's looking in this direction, but then suddenly it changes a bit, so it repositions towards 150 degrees. So it goes from here to there. So you might wonder, yeah, who cares? I care. So, for example, if we look at the red area, then we know that the bird is overall looking in a 30-degree angle. So it's looking in this direction, the body is positioned like this. But then we see that when he catches an insect, it's in certain degrees next to the tree. And then again, yeah, who cares? I care. So what we can do is go to these positions. We know when the bird was there under which conditions. and then we can start to look at these cloud data, or sorry, the light environment data. So we can actually try to understand what kind of light an individual is using under which conditions. He is finishing up his manuscript and actually forgot to put some of the results in it. But anyway, what he finds is that when there is a moon available, nightjars will usually perform the light of a rising or setting moon. When there is a full moon, then they don't really care. The night is so bright that they can look in any direction. And when there is no moonlight, these nightjars, they will tend to orient themselves towards bright sections of the sky that are influenced by skyglow. When it's an overcast night, then you more or less have the effect of a full moon night, that it's become so bright that these birds can look in the direction that they want. And then the final thing that is one of the big mysteries for us is that at the end of the season we catch many juveniles, even though that we perform quite some field work during the season and we monitor most of the nests that we have in our sites, but still there is a big influx of individuals. So what we will try to do now in the upcoming years is to try to understand how juveniles within our own population, but also the individuals that come into our populations at the end of the season, who they are, what they do, where they go to, and how, for example, the light environment or the habitats, different habitats in our study sites might have an impact on natal dispersal. So what we will do for this, so this is Belgium again, with a slightly different view on the light pollution map. So we have the bright area in in the northern part, the dark part in the French-speaking part of Belgium. And we are currently setting up a MOTUS network that covers the entire breeding area of the species in Belgium. And we hope to understand how juveniles might use the area pre and post-migration. From the data that we have nowadays is that we understand that juvenile dispersal, or at least the individuals that we are able to recapture, is approximately 11% of the juveniles that we have during the season. And that on average approximately 7 kilometers, but we also have a few individuals that go to completely different sites. So to be honest, we don't know what is going on. And we hope to understand this during the next 4 years. So this was a summary, and thank you for your attention. And if you have any questions, then I'm very happy to answer them.
So thank you, Ruben, for your super interesting talk. So I'm sure there will be many questions about this topic. Yeah.
Thank you for the excellent talk, Ruben. I was really impressed by metabarcoding you've done, but as I understand, the metabarcoding you did wasn't done in conjunction with studies on the skyglow impact. No. I was wondering, looking at the plot of activity of the birds in the dark sky and more sky glowy parts, are you planning to do a metabarcoding in conjunction with that? Because so I'm working with artificial light at night IGB group, and we know that on spotlight pollution, we didn't look at sky glow, does change the composition of insect community. Yep. So it might mean that they might need to do more tries, for example, because they get in small mosses or beetles.
Yeah, that's for us that super interesting question, and for us that's also the big question. How does sky glow affect these activity of moths in foraging sites, but also in the breeding sites, and also how does it change the composition? They use a specific group size of individuals, of moths. But yeah, at this point we have no clue. The study on the foraging, the prey availability was done 2018, I guess, and we started to come up with the idea of skyglow 2 years ago. So yeah, So we have Richard now, and then we will work on natal dispersal. And the next step is to find someone that can work on community dynamics. So, and then that's the goal of understanding how skyglow affects activity of moths and also activity of nightjars. Because that's also we see that they perform more flycatches when the skies are brighter, but it could also perfectly be that they need to perform more flycatches because they in catching the prey. And that's something that we cannot grasp at this moment. We can just hypothesize.
Thank you.
You're welcome.
My question was related. So my question was related to the first one. It's because you have now seen that the birds are more active when they stay down. But then, do you have any measure or any idea of how this is affecting the business of the species? I mean, no.
Not yet. Yeah, that's part of Richard's work. If you talk about fitness instead of in terms of reproduction, or
or maybe any measure of the body condition.
Yeah, that's one of the flaws of our study. We don't measure body condition. Yeah, we have weight, of course.
How do you check that?
Weight, stomach content, but we didn't relate this yet to, for example, differences in light conditions in the different study sites or stuff like that. We have not looked at that yet. But yeah, that's something that we are planning to do in the very short future.
Yeah, maybe just using the way that is very basic, but maybe.
We actually measure a lot. We measure weight, stomach content, stuff like that. But yeah, it's always usually it's 1 or 2 measurements per individual during the whole season. So it's not that we measure it. We don't capture the individuals 3, 4, 5 times per season. So we don't have, for example, stable measurements of conditions per individual. That's something. But yeah, to be honest, with this whole skyglow thing that exploded in my face 2 years ago, there are so many things that we should do, and we just haven't done it yet. But that's one of the things that we want to understand, how it affects individual fitness, but also reproductive success, for example. And that's something sorry if I continue. I just think it's interesting because it's not only affecting the nightjar, but also everything else around it. So predators, for example, can also increase their activity and reduce the reproductive success of nightjars, for example, because there's more predation, and it's a lot is going on. Yeah.
Okay, thank you very much for the talk. It was very, very interesting. And there are lots of questions. And I'm following up a little bit on what Victor was asking, because you were commenting on higher number of flights on nights with more light. So then seems that it's more difficult to catch something, that they need more energy, or what happens the following day. Maybe after one wonderful night, the following day doesn't matter. But I don't know, it's very interesting, but it seems that a conclusion of the work is that somehow night pollution could be beneficial for nightjars, in the sense that maybe easier to find food. Do you have any data suggesting changes in the distribution taking into account light pollution?
How many days do we have to discuss this topic? It's one of the most tricky questions that, to be honest, it's also one of the, when I applied for a project on this, it was rejected once because the reviewer said, we cannot fund this because you suggest that light pollution is beneficial for a species and light pollution should be negative. Eventually someone else reviewed it, it was successful. But I maybe in the short term it could be beneficial for individuals, but I don't know how sustainable this is. It's also going at such a rapid pace. I think these birds are flexible to a certain extent that they can rapidly change their nocturnal activity to, for example, increasing their time that they spend foraging, because otherwise they would be resting. But at the same time, I would expect there is a certain asymptote that they work towards, that they don't need to feed constantly. They also have a limited stomach content, I assume. And we just don't know how it affects them in the long term.
But maybe there is data on the hatching success, on the reproductive success of individuals living on environments with different amount of light.
Yeah. So we don't have information on capturing success. I mean, capturing. What
Not
do you
capturing the productivity, number of chicks that are.
Yeah. So that is something we have now. But again, we didn't look at it yet. And to be honest, I don't. This is anecdotal, so we have looked at the data, but we have not analyzed anything. So what I say now is not super scientific, but it's based on the experience. I don't have the impression that the brightest sites are the ones that yield the most or the highest reproductive success. And I even think there is some kind of response curve that you have bright sides are not good, but the dark sides are also not good, and maybe there are different processes acting against having success in bright sides and dark sides, and there is a sort of optimum that a bit of extra light can be beneficial, but extremely bright sides not, and dark sides neither. So yeah, actually, I hope to tell you in a few years, to be honest, what is happening. Yeah.
Thank you for your talk. It was really, really interesting. And correct me if I'm remembering wrong, but you had a few individuals in Mongolia and the individuals in Belgium, and you found a difference in activity, right? Mm-hmm. But I saw on the map that they were relative. Do those differences remain, or do they fully disappear?
They disappear, yeah.
Yes.
Yeah.
Okay.
So we compared the birds during the breeding season, and then the birds had higher activity in Belgium relative to Mongolia, and then compared to the same individuals during the wintering site, and then these differences, they just disappeared.
Okay, yeah. So right now it's behavioral. Are you looking into or expecting at some point that those changes will carry over to overwintering sites as well? Or is that something that you haven't thought about or haven't had time yet for?
That's something that we think about every day. So I have the PhD students now, and currently we're setting up a postdoc project with my former student that works on migration. So his goal is indeed to understand how light during the breeding season might translate into or carry over into later periods of the year, so at the stopovers, but also in the wintering sites. And I secretly hope that we find differences between light and dark populations, but that there could be some kind of reset during winter, and that they synchronize again, and then, but yeah, that's hoping and dreaming, and but yeah, that's something that we are thinking about.
All right, thank you very much.
Thanks. Just a small remark, as Ruben is my supervisor, I would want to follow up on what you said. I think it's inherently human to think if something is good or bad for the animal or for the study system that we have. But I think the truth is that they really just take the advantage of the opportunity they have, because you have something which is deeply rooted in their evolution and behavior, that you have this 28-something days moon cycle, that they know it's always regular. But then they have to be highly plastic when it comes to clouds, for example. So every night the situation is different. So they have to adapt their behavior based on the cues they get from the environment. So Then if they get an additional source of light, it simply means for them that, okay, it's as if it was a bright night without clouds, so they can adapt their behavior to that. But yeah, we indeed have to prove it that they can perceive it that way. So just the small remark.
Just a very small remark. How to counter the most typologically, box-headed reviewers. Yes, they might benefit in the short term, as Richard said, but they're gonna run out of mosses at some point, because as one of our researchers show, bright light pollution inhibit egg disease in many moss chrysalises, and it's not great.
Yeah, some of the things that the hypothesis or the ideas that I've been playing with, and as long as we don't measure moths, then I will not this time, short term, it might be beneficial for individuals, but maybe only because they now have more time to look for the fewer insects that are available. And we don't have the long-term data on moths, so we...
so Belgium doesn't have it, but Netherlands seem to lose 60% of mosses and 45% of caddisflies in the last 25 years.
Yeah, so that's why I think maybe they might, they just have extra time now. And maybe the only beneficial part is that they just have more time to look for food, and maybe that's why they do it good on the short term, but we don't know how this can collapse in 10, 20 years or so.
Anyone else? Any questions? I'll check. I don't think there was any online. No, there's no questions online. So I think okay. With that, then I think let's wrap it up there. Thank you again, Ruben. Let's give him a round of applause again. Thank you very much. And we'll see you next week for the discussion group, I think. Excellent. Thank you very much. It's nice to be back with so many people. Listo.